Kedves rektor úr, államtitkár úr, professzorok, professzorasszonyok és vendégek, nagy öröm itt lenni. Nagyon szépen köszönöm az egyetem vezetőségének, hogy nekem ítélte ebben az évben a Semmelweis Budapest Awardot. Itt voltam diák, 24 évvel ezelőtt fejeztem be az egyetemet, és azóta próbálom az itt tanultakat gyakorlatra váltani és továbbvinni az orvosi egyetem eszméjét. Tényleg egy nagyon megható egy olyan díjat kapni, amit az ember attól az intézettől kap, attól a közösségtől kap, amelyben a gyökerei, ahol a gyökerei származtak. És mind a Szent István, mind ez a díj talán nagyon közel áll a szívemhez. Az összes többi díj, amit kaptam, a szakmai, ezek szakmai és a emberi és az eredeteim, a gyökereimről szólnak. Today I would like to talk about something that I'm studying since many years, and that is to restore vision. Our vision starts with a video, and that video is captured by the retina, and from here information flows to higher brain centers, and for us humans the LGN is the most important. Then information arrives to the primary visual cortex, and from this it moves to a large number of cortical areas, and indeed in humans in 50% of the cortical areas you can measure visual activity. So vision is really important for us. In the last 24 years I have studied how information processed in the retina, the LGN and the cortex, and we had a large number of collaboration with Balázs Rózsa using new imaging technologies to show how an image enters the eye and gets more and more complex as it's transformed by its route. Today I will not talk about this basic research, but another aspect of my inquiry, namely turn understanding into therapies. The retina is only a tiny part of the visual system, yet almost all diseases that lead to blindness originates from the retina. The retina is a computer. It is a computer that acquires one image that enters the eye, and it creates around 30 videos that exit the eye via the optic nerve towards the brain. And each of these videos is pulling out a different feature from the visual scene. This is an elementary language, how our retina describes the world. Our brain is unable to see the visual world, but it sees these retinal videos and real-time guessing what is out there based on these videos. If we look inside the retina, what we see, and this was already seen by the famous anatomist Ramón y Cajal, is a lot of different morphologies. Cells come in different shapes. And Ramón y Cajal had a major observation, namely every morphology can be repeatedly found, hence the origin of an idea of a cell type. And this concept of cell type dominated the last century our understanding of the functional architecture of the retina. We and others worked on to figure out how the information from photoreceptors then goes through these different circuits, then to end with ganglion cells, then talk to the rest of the brain. However, at that time, there was no connection to medicine at all. But 10 years ago, something major happened. Few laboratories, including my one, realized that these cells not only have unique morphology, but they have unique gene expression patterns. This was the time when we started able to record gene expression in individual cell types, and we have seen a complete unique gene expression pattern from cell type to cell type. And the reason it connected ophthalmology and with research, because we knew from ophthalmology that every retinal disease is cell type specific. There is no disease when all the retinal cells would die. And the two came together suddenly because now we understand that disease is cell type specific, either in a monogenic disease that gene is only expressed in the particular cell type, or the vulnerability of cell, the genes that make up the vulnerability gives a specificity. Recently, so that led to the understanding and inquiry into cell type specific disease mechanism. And finally, in the recent years, we and others, as we started to develop therapy, realized that the cell type concept is also important for therapy development, and no one today is developing gene therapy without targeting cell type, because otherwise it's unsafe for people. We can induce a disease if you go non-cell type specific. Now, the details of this retina is highly conserved from zebrafish to humans. However, interesting, so the general architecture is conserved from zebrafish to humans. However, the details are quite different. And these details turn to be so important for medicine that almost no animal models are reproducing the human phenotypes. And if we start to develop therapy in mice, we will never arrive to humans. So recently, we and others realized it's at utmost important. This is what also Arnold Szabó told you about, to understand the human retina. And this is what I will talk about today. I will talk about the function, how we understand the functional architecture of the human retina, how we can study disease mechanism even in rare diseases in a human retina, and then I talk about therapy development and some new avenues how to solve current problems with therapy development. The most fundamental problem we have faced many years ago is that how can we record function from a human retina? The human, we cannot, we can look into your eyes, but we cannot record at single cell resolution. And the insight came from multi-organ donors. The idea is super simple. We can take retinas for multi-organ donors, then put them in a dish and record from it as we can record from any animal species. Usually for one day, we can keep a retina fully functional. However, as 8 years we spent on it, we never get a light response. And we realize that the secret is the perfusion, that if we are later than 5 minutes after the stop of perfusion, we lose light responses. Therefore, in Basel, we worked out a legal and a medical team that within 5 minutes after a multi-organ donation, we can have a retina in oxygenated solution, and we can put it on a chip. And this chip was developed in Basel with my collaborator, Andres Hirleman, that has 30,000 electrodes, so we can put the retina ganglion cells down and project images on it. And then here you see our first human retina as it's spiking. This is not yet light-evoked spiking. We can turn on the light, and here real time we can record from 1,000 human ganglion cells responding to a flickering light. Indeed, we have 16 hours to record from this retina, and many, many terabytes of data we can collect, almost completely describing what the human retina is telling to the human brain during visual information processing. But having this in a dish, this is, of course, we cannot make medicine by understanding retinal processing. However, in this case, we have a healthy human retina in our hand, and what we can do we can dissociate to single cells and create a single cell gene expression pattern. It's a new technology in which we can take 100,000 individual human cells from this functional human retina, and each of them we can measure the gene expression. We can measure several thousand genes, and from it we form a gene expression map. In this map, every dot is a cell, and 2 dots are closed if the gene expression is closed. It's a projection from a high dimension to 2 dimension. What you see here, these clouds representing cell type. And averaging this information, you can get the cell type gene expression for every human retinal cell type. And now really comes the important thing for medicine. We can turn this into a map in which we take human disease genes. Every row here is a human disease gene from a genetic disease, and we can ask which cell type it is expressed from these maps. And so we obtain the world's first fully described human organ in which we can map any disease gene to its cell type. Why is it important? Because now we know where the disease originates and which one we would like to target for therapy. However, what we cannot do with these retinas, we cannot study genetic diseases, because the likeliness that a disease that is 1 out of 10,000 we will get a donation is very close to 0. Therefore, we need completely new technologies to create human retinas, to build human retinas in a dish, that we can study any human genetic disease. And these investigations, again, started around 10 years ago in my laboratory, where we started from skin biopsies, where we can fibroblast cells, or today we can also do from a blood donation from lymphocytes. We can turn these fibroblasts into induced pluripotent stem cells, and using a combination of few factors, we can induce the formation of optic cups in a dish from this cell mass. Once we remove these optic cups, these become a so-called retinal organoids, and around 80 days of age, these organoids start to develop towards the retina. In one side, you can see the photoreceptor precursors, the other side, the ganglion cells. And this is all this technology so far was coming from Japan, from the laboratory of Yoshiki Sasai, but the field could not reach the mature retina state. And we have worked several years to try to add different factors, but we as others couldn't succeed to build a beautifully 5-layered human retina that Arnold showed you before. And the insight came around 3 years ago, when once I was extremely frustrated of going nowhere after 5 years. And when I'm frustrated, I usually go back to mathematics. That's my hobby still. And immediately became clear to me on the day that what we are doing is really stupid. And the reason for that, because the growth of a retina is a nonlinear system. As any other nonlinear system, what we know that what is important is the initial condition and not how you go from one point to another one. We also know from nonlinear systems that if you change the initial condition, you can get qualitatively completely different outcomes. A qualitative outcome would be a normal human retina as it looks, a 5-layered, and another qualitative outcome would be this nonsensical tissue that doesn't look like a human retina. So the idea was that instead of adding all sorts of factors, try to look for clones, iPS clones, that might have the ability, they might have the initial condition to grow to 5-layered human retina. And once we had the idea, within 1 year we had the solution that in 200 days we could develop in a dish a human retinal organoid that had a 5-layered, including the synaptic layer, the photoreceptor layer, autoplexiform layer, intranuclear layer, inoplexiform layer, and the ganglion cell layer. These organoids also grow the light-sensitive apparatus, the outer segments. And they had almost all the human retinal cell types that we see in a normal retina. They had the cone mosaic growing. They have the rods, exactly how they arrange in the human retina. Even we could find subtypes of cones responsible for vision, for color vision, like the blue cones and the red glint cones. And we can go on and use markers to show that the organoids have the bipolar cells, the horizontal cells, the Müller cells, and many other cell types. Moreover, these cells are connected using by synapses, and you can see here an electron microscope era of a ribbon synapse grown in culture. Now, these human retinas, of course, we didn't know at that time what is the age of an organoid compared to an age of a retina in the fetus, in the mother's tomb. And then we have done gene expression studies, and remarkably, we found that the growth rate of an organoid retina in a dish is identical to the growth rate of the retina in the mother's tomb. Therefore, a 6-month-old organoid equivalent of a newborn baby's retina. Of course, what is important now is to know which diseases we can study in these organoids. And this we can determine by again using single-cell gene expression in mature retinal organoid and compare to them to the adult human retina, and now comparing disease maps in both organoid as well as the periphery, as well as in the retina. So you can already see that the two patterns are quite similar, and our own gene-by-gene comparison tells us that in 70% of the cases, we can see the disease gene in the right cell type in these organoids. Now, what's the beauty of organoids is that you can grow them from any patients. From any of you, we have even grown them from 80 years old patients. I can grow a retinal organoid, but much more importantly, we can take our control organoids and edit in the mutations using gene editing technologies. So in these years, in my new institute, we are building essentially all diseases and storing for large-scale European studies that in the freezer, we have almost any human genetic disease that we can take an organoid, and if therapy comes available, we can test therapy on them. So this is one way of using these organoids is to edit in or take patient-derived material and build organoids to study different diseases. But there is another major use of it, and namely study the role of environment. Many of you know, especially ophthalmologists, that only a few percentage of diseases are genetic diseases. The majority, for example, we don't know the reason. For example, glaucoma, we know that the pressure is high, the ganglion cells are dying, photoreceptors are not dying, but we have no clue why the ganglion cells are dying. Or for example, in retinitis pigmentosa, people believe that although the cones, they don't express the mutant gene, but they may die because the low glucose are on them. But how can we test them? And organoids provide a way to test them, since here we can create an adverse environment that leads to damage of a specific cell type. We can observe the damage and screen compounds to prevent the damage. So the idea that if we are able to make thousands of millions of organoids with this technology, then we can replace classical pharmaceutical screening on cell lines going to the right cell types in organs. And this is recently achieved. Now we can make, by two inventions, we can make essentially unlimited number of organoids. We created recently several thousand, but the technology allows us to make several millions. And now we're just robotizing it and put them into multiple plates and start screening. So what are we screening? And I'll show you one of our first nice insight. How can you screen? First of all, in these organoids, we can take a virus that Arnold briefly introduced, and we can, with a drop of virus, we can specifically label essentially 100% of the cone photoreceptors in these retinal organoids. And then, since the cones are labeled, we can see on each well, we can see these fluorescent retinal cells. Each of them is a cone, and we can take away glucose, and over 2 days, we can see as the light goes on, each cone is dying over time. So this one can do massively parallel imaging and compound screening to learn how can we prevent the cones from dying, for example, in case of retinitis pigmentosa. Finally, understanding disease is not the same as developing therapy. So I like to finish my talk with discussing what's the route to develop therapy and how we go from understanding from a human retina into a living human eye. And the miracle really started with this little virus called the adeno-associated virus, or we call them viral vectors, AVs, since gene therapy has been in the news since many, many years, except there was no safe viral vector. And what is amazing about these AVs, that they are safe. Now, we and others injected into the human eye, in other parts of the human body, and we do not see adverse reaction. These are not viruses as you imagine viruses. These are all synthetic. We make them in the laboratory. They don't replicate. They are just little balls that can enter a cell and deliver a gene. They have a capsid, so it's a protein ball, and inside is a single-stranded DNA, which has two important regions. One is the gene you want to bring in, and the second one is what we call a synthetic promoter. And a synthetic promoter is nothing else than as a 1 kb or 500 base per long DNA sequence that we like to use as a postal address. The AAV will enter every cell, but we only want to express a gene in a particular cell type. Now, how we find these promoters is a complex problem, because in principle, if you have a sequence of 1,000 long, it's 4 on the 1,000th combinatorial you have. It's impossible to figure out. So 8 years ago, my laboratory started to bid on all sorts of partly mathematical, partly heuristic approaches. And the nice news was at that time when I told people that I will make synthetic promoters in AVs, everybody told me we lost our mind. It will never, ever, ever work, because you need the genomic environment. And it's very encouraging when your colleagues are saying that, because the game becomes exciting. And the idea was relatively simple. That is that we were aware that we were unable to perfectly predict which cell type to target, or what we initially wanted to guess, what is the probability that we can target a specific cell type. So I will not go over the heuristics, how we create these sequences, just to tell you that we have made 250 different adeno-associated viral vectors and went with different synthetic promoters and went to screen in mice, non-human primates, and together with Arnold in human retinas. And I like to really point out that the key technology coming from Arnold here, and initially by Serge Picot, since Serge was able to, at that time, keep human retinas alive for 3 weeks, and Arnold, that is not possible to screen for viruses in 3 weeks, but Arnold made it possible to elongate it to like 14 weeks, and now we can screen AAVs in the human retinal cells. This later became very important. What we've learned is that if you target a cell type in mouse, the probability that you will target the same cell type in humans is close to random. So it's totally useless to start to develop gene therapy in mice. In primates, actually, the hit is not so far. You're at the 80% probability. But in these human retinas, we could cut the human retinas to small pieces, and that's old Arnold's miracle, how he does that, put on the viruses, and there we really started the first hits. For example, this promoter, ProA7, completely specifically targeted cone photoreceptors, and up to 80% of them. Here you can see that we can target rod photoreceptors much less efficiently. You can see we can target also horizontal cells, and we can go on, and that target one by one more and more human retinal cell types. Now, 250 allows us to target around 20 different cell types. We have much more in the retina, and now we in an institute, we are scaling up to several thousand viruses. So at the end of the day, we would like to have a library for gene therapy that anybody in the world basically can pick a virus and say, I have a gene, I want to bring it to this cell type, and then you just put in a gene and go, and it worked in human retina because that's where we tested it. Now, I like to give an example how we use these viral vectors, and this is what we call a cell type targeted optogenetic therapy. And initially, we started this therapy in retinitis pigmentosa. We heard about this disease. It is a disease of rod photoreceptors, and rods are dying, and you lose low light level vision. However, we humans do not care about rod vision. Indeed, we always turn on the light to see colors, so we almost essentially always use cone vision. The mutated gene usually is not expressed in cones, but for unknown reasons, the cones are bystander and lose at the end of the game the light-sensitive outer segment. At that time, the patient goes blind. And the idea that we had around 15 years ago is to target a cell type in the retina with a simple light-sensitive channel to make artificial photoreceptors when the normal photoreceptors are lost. This is an extraordinarily simple idea because at that time, people from Max Planck Institute and Bomberg published a light-sensitive channel from bacteria that was relatively simple express. And then around 14 years ago, we made a sketch which cell type we would like to target for therapy, either the remaining cone cell bodies, this is the best, or if the cones are not anymore there, the bipolar cells, A2 hemacrine cells, or ganglion cells. And although it was easy to make a drawing, it took us 15 years to arrive to clinical trials. And this ganglion cell targeting, which is by far the simplest and giving the most primitive vision, now is in clinical trials. And there are at least 5 to 6 new companies that now strategizing in these different other cell type targeting. In our institute, we are moving for gene therapies targeting cone photoreceptors. And again, it took around 15 years to move initially from mice to human retinas to test in primates, and now finally we are in patients. Now it's not only an injection to the eye, we also need we need a goggle for this, because the bacterial sensors are only sensitive in a particular wavelength at a particular intensity domain. So this was made by a company called Genzyme Biologics. We have founded in Paris. It has a video camera watching the outside world and a projector that projects information onto the retina, and in between the computer that we can program it to perform different image computations. Now, as every approach in medicine, each method has a problem. The problem of optogenetics is that you can only use it in completely blind patients. If the patient have remaining vision, this will not work because we have a strong light projected on the retina. And the completely blind patient, the number of them is relatively low. So we need new technologies. For example, in macular degeneration, the periphery is intact and we cannot project high-intensity light. And macular degeneration is by far the largest photoreceptor degeneration compared to its pigmentosa. So recently, we started to think about what can we do to not to interfere with the remaining vision, to regain vision in a part of the retina, but leaving the rest normal. And the idea was relatively simple, that instead of using normal light, we want to make near-infrared light sensors in the eye. Now, optogenetic tools are unable to sense near-infrared light. That would cross the boundaries of physics for them to do. But we now again turn into nature. Is there any species that can detect infrared light? And sure enough, if you look around, nature can do a lot of things. And it comes up with these beautiful animals. One is called vampire bats. Another one is different vipers or different snakes, for example, pit vipers. And these animals, they have an eye, but they also have an other organ that detects infrared radiation. And in their brain, the two images are overlaid to allow capturing of prey. So of course you have an advantage. If I am against a pit viper and the pit viper can see me when there's almost no light, I will lose. And that's their advantage here. And the question what we want to do is to learn them how they do it and artificially create a system that we can create near-infrared sensitivity. Now these animals, they actually don't detect near-infrared but infrared radiation. They use temperature-sensitive channels like TRPV1 and TRPA1 channels. And the problem of directly simply inserting a temperature-sensitive channel in, for example, the remaining photoreceptors is that if you would show infrared light, what would result is to cook the retina and you will be even more blind than you started from. So that's not a good idea. Rather, what we want to do is insert a temperature-sensitive channel, but only heat it locally. And this idea essentially came in one of my Budapest visits 6 years ago. I was in one of the hotels, and my father was very ill. I didn't know what to do, and I thought that when I don't know what to do, it's better to invent something. So this is essentially was all happening in that hotel, where I realized what kind of system we would like to build to allow near-infrared light sensitivity. And it's really, really simple. We would express a heat-sensitive channel, a TRP channel, which we engineer with an epitope that we can come with an antibody and a gold nanorod against. What the gold nanorod does is the following. The gold nanorods are little antennas that are inert and can capture near-infrared light depending on the length to width ratio. We can tune it. And when they actually capture that, they radiate heat very locally. So if I bind them to the channel, they will heat the channel up molecularly, open up, and create a current. And indeed, I don't go through a lot of science. This paper is under review now in Science. But we just want to end that with using this technology and targeting to cones in a human retina, 9 weeks post-mortem, we can completely resuscitate the human retina now that it responds to light. So we targeted the near-infrared light sensitivity to photoreceptors. They start to respond. They instruct the bipolar cells to respond, and they instruct the ganglion cells to respond. Now we have a human retina in a dish that is seeing as if we would be seeing in patients, except it's 9 weeks after death. So these retinas, not only important as a proof of principle to develop therapy, but now we have a tool to study human vision for many, many weeks. What we can do with these retinas is simply give them a drop of AV to bring back the light sensitivity to cone photoreceptors, and another drop of AV that has a genetically encoded activity sensor to another cell type. And we can study the computation between photoreceptors and that cell type. And since we have a large library of a cell type targeting vector, we can basically go from vector to vector always through dorsal-viruses and study the information transfer from cell to cell. Finally, in many cases in neuroscience, when you are recording from a brain region, it's very important that if you see a cell, you can somehow bring a gene in that particular cell, instead of bringing too many cells. So last a few years, we developed a new technology that allows one to pick a cell, any cell, anywhere in the body, and simply touch it and bring a gene into it. And that we call viral stamping. This is a technology we developed with Daniel Müller and recently published in Nature Biotechnology. We take a glass needle that you pull, it's extraordinarily simple technology, coat the tip with some chemistry, and then take a jar of virus and put this in. When we pull out, the viruses now shown in electron microscopy needle rabies viruses are attached to the tip of this glass needle. And we adjust the forces such that when you put the needle into a solution, they don't come off. We can, however, touch a cell, and then after touching the cell takes up the virus and expresses any gene of interest. Moreover, this technology is easy to generalize. You can take a jar with many viruses, and you can put in your little glass needle, put on a cell, and then you can bring in many viruses. Here's an experiment where we now take two viruses attached to the needle, and then a cell becomes red and green because the two different viruses had a green and red fluorescence protein, so it becomes yellow. Now, at this point, I like to end and just briefly summarize that vision really starts in the retina. It's a very complex route, and the percept is really formed in the cortex. Nevertheless, the retina is the part in a human where we can inject different drugs, viruses, and it's extraordinarily lucky that it turned out to be that most visual diseases are indeed in the retina. And we and others in the field are believing that with the advent of gene therapy and the technologies that can study human retinas, both built in a laboratory, like a form of organoid, or keeping them alive, as Arnold showed you, and technologies that Balázs invents to image them, we can have a new chapter in ophthalmology where we can directly develop better therapies. So at this point, I'd like to say thanks for a few people. One is Hendrik Scholl, the co-director of IOB, the new institute, who is a clinician. He's the chair of ophthalmology, whom we are bringing several therapies to clinical trials. Norbert Spirig, our director of operations, that helps or allows us to do science instead of focusing on administration. And I'd like to say thank for 4 people who have been instrumental of me being able to do science instead of doing other things. Tamás Szikra, who has been a head of scientific operations at our institute. Dorothy Sears, who has been my grant officer since 15 years. Brigitte Gaucher, with whom I started my laboratory as technical assistant. And most importantly, Elsa Ziegler, who is the head of my office, who is also here. And he's also basically running myself and allowing me to concentrate on science instead of doing things which are absolutely horrible, which is administration. Thank you very much.